What the heck? Okay, can somebody confirm you can hear me?
Yes.
Thank you. Make sure I'm running. Looks good. Get that out of the way. Oh, I can't move. There it goes. So today is January 11th, and we're going to be talking about Lab 1, the ubiquity of microorganisms. Let me remind you that you need to get Lab 00 worksheet in by 8 a.m. Friday morning, or else you will be dropped from this class. This is the only attendance I'll be taking. I'll be taking attendance by your turning in Lab 00 worksheet. Let me also remind you that the online plagiarism quiz is due this Saturday, January 13th at 11:59 p.m. and that you must score 100% on this plagiarism quiz. If you do not score 100%, then you will get 0 points out of 100 on your infectious disease project. Also, what's due at 11:59 p.m. on Saturday, 1/13, is this worksheet for Lab 01. Now, if you take the online plagiarism quiz and you don't get 100%, you may take it again. Rarely have I had a student required or had to take the quiz more than twice. Usually 2 times will be enough. In fact, most students get it right 100% the first time. Let me see if there's anything. Oh yeah, there's one other thing I need to talk about, and that is we have for the infectious disease research project a file on campus called Project Instructions. I'll show it to you now. Come down below week lab 1. I believe it's week lab 2. There's the infectious disease research project. You should open this up and read the instructions for the infectious disease research project. I'm going to talk about this, I think, in our next lab. Yeah, the infectious disease project. All right, so let's begin Ubiquity of Organisms, Lab 01. Any questions about anything? Got that open someplace here. Lab Module 1, Ubiquity of Microorganisms. As with all of the labs, there is some reading from the textbook. Not very much. You have a couple of pages there, that's 4 or 5 pages on culture media, and then about 1 page on obtaining pure cultures. There will be some supplemental material in this lab. You'll be watching some video clips. The learning objectives for this lab is 1, know that microorganisms are almost everywhere. 2, describe the purpose of culture media. 3, describe colony abundance and colony morphology. 4, define the following terms: inoculation, ubiquity of microorganisms, what's something that's sterile, what is isolation, incubation, in vitro, a culture media, and of a culture media, we have different types. There can be a broth, that's a liquid media, and then you can have an agar, that's a solid culture media. You can add agar to a liquid culture media to make it a solid. You can add a few other things, mainly gelatin and starch, to make it solid. But most of the time when we make a media solid in microbiology, we're adding agar. And then a slant and a deep, and I'll talk about them later. What's something that's fastidious? What's a non-selective media? What is ubiquitous? And what is a colony? So microorganisms are organisms that are too small to be seen with the naked eye. These do include viruses and prions, even though they aren't cells and they aren't technically alive. We'll also include helminths in our studies of microbes, even though the adults are not microscopic. However, the infective stages of the worms are microscopic. The phrase ubiquity of microorganism refers to the fact that microorganisms are essentially everywhere. Something that is sterile is something that is free of living cells, including microbes. So when you come into a lab and you get a sterile media, it will be autoclaved to ensure there are no microbes in the media. In this lab, we'll investigate the abundance and diversity of microorganisms in common environments, and you'll also learn some basic lab protocols. A culture media, also known as a growth medium, is a sterile laboratory preparation that contains a variety of nutrients that are required for growing microorganisms in the lab. Meaning the culture media has all of the nutrients for microbes to grow. A culture media may be a liquid, these we call broths, and they're usually used in a test tube. We can add agar to the liquid medium to result in a solid medium, and these are often grown in plates, but they can also be grown in a test tube, or the media put in a test tube. Here's a video you can watch on preparing pour plates, and you should note how the culture medium needs to be sterilized prior to its use. Here's a question for you. Why must a culture media be sterile prior to it being used in a microbiology lab?
So there's no cross-contamination?
Yes, so there's no contamination from other organisms that you don't want to study. So there is a petri dish, the lid and the base, and you pour media in it, usually solid, to obtain your media in this. And usually we have a solid media in a petri dish because if it was liquid, it would just be too easy to spill it out of the petri dish. Our liquid media that we call broths are normally grown in a test tube, and here we have a broth. Here we have test tubes with solid media. This one here is called the slant for obvious reasons. It forms a slant, and that where the agar goes across straight, we call these deep, or an agar deep. We're going to talk about a variety of different types of media in microbiology. Today, we're only going to talk about the general purpose media. These media are non-selective in function. They're usually of complex composition, and they support the growth of most non-fastidious microbes. A fastidious organism is one that does not grow easily, and to get it to grow in a general purpose media, you have to add specific additives to the media to get the fastidious organism to grow. So fastidious means it's difficult to grow. And an example of that would be Streptococcus pyogenes, which causes strep throat and a slew of other human diseases that are actually fairly rare in the United States now. But they used to be fairly common before the introduction of antibiotics. I think it was things like scarlet fever and, oh, let me see, I used to know this. Rheumatoid fever, maybe. Anyway, Streptococcus pyogenes is fastidious. It will not grow on a general purpose media. You need to add specific additives to the media before Streptococcus pyogenes will grow. Usually, we grow Streptococcus pyogenes on a blood agar plate because the red blood cells supply the ingredients that Streptococcus pyogenes needs in order to grow. Any question about any of that? All right, I'm not hearing anything, so let's talk about the general purpose media. The first I'm going to talk about is actually number 3, nutrient agar. It's a simple agar that is simply beef extracts, meaning ground up dried cow, and digested protein that we call peptone, and then sodium chloride. I think those are the only 3 additives other than obviously water in nutrient agar. Tryptic soy agar, another common general purpose media. It is also abbreviated TSA, and I guess nutrient agar is abbreviated NA. Tryptic soy agar has protein from soybeans in it. And then nutrient agar with yeast extract is a little richer than nutrient agar. It is nutrient agar. You just add yeast extract to it, and it's just a little richer. Any question about general purpose media? All right. These are the only media we're going to talk about today, but we will talk about other types of media later on in the term. Whenever you're growing something in the microbiology lab, you have to have appropriate labeling. The Petri dish or the tube should be correctly labeled prior to use with the following 4 or 5 items. You must put your name, the date, the type of medium it is, like NA for nutrient agar with yeast extract, the inoculum, what you're going to grow on the culture media, and that could be something like E. coli. If it's something you don't know entirely what it is, just write how you got it. Like if you're growing organisms that you got from opening the plate and exposing it to air, just put air exposure. And then the incubation temperature is optional. You may or you may not add that. To label on a tube, you need to write on a piece of tape and then attach the tape to the tube. On the plate, you write on the bottom of the plate. And there's a reason for writing on the bottom. And that is if the bottom and the top get separated, the cells will be on the bottom of the plate. And so you write on the plate, bottom of the plate, to make sure you know what's in the plate. And I once worked in a lab where in the lab next to me, there was a lady, a young lady too, who was working with a bunch of Petri dishes. It was at least 100, maybe a couple hundred. And she all had them all on a tray. And she was moving them from one place to another. And while she was moving, she tripped, spilling the Petri dishes all on the floor so that the tops got separated from the bottom. And then people tried rescuing what they could, and they noticed that they had written everything on the lid. And so no one knew what was in the bottom of the plate. And so the whole experiment had to be tossed and then repeated again. And if she had written on the bottom of the plate, that wouldn't have happened. When you're writing on the bottom of the plate, you should write along the edge, because if you write in this region of the plate, you'll obscure what you may want to look at, and you'll make it more difficult to look at, certainly from this side, the bottom, but also from the front, because the writing will come through the auger. You should put the name of the person, what they're growing, E. coli, the date, the media, TSA, and then the temperature you're growing it at is optional. And inoculation is the placement of microbes into a sterile culture medium. So when you're growing something, you will inoculate it. And then you take that culture media and then you put it someplace for incubation. Usually you put it in an incubator. So an incubation is placement of the culture media that's been inoculated in an appropriate physical environment for a period of time to allow the growth of microbes. The fancy incubators where we do the incubation can control a number of things, including oxygen, the level of carbon dioxide, the temperature, the humidity, and of course the time. And this is a complex incubator here, controlling all of those functions. In our microbiology lab at Clark, we have simpler incubators, and they only add temperature. You can set the temperature, and you add a dish that you put water in to give humidity to the growth. And the humidity is helpful because otherwise the plates in the dish will dry out faster. So you want to add humidity to what's growing. And then the only other thing the incubator does is it regulates the time, and that is you put it in to grow, and then you pull it out to stop the growth. Well, not stop the growth, to slow the growth. Any question about any of that? All right. When you're putting plates in an incubator, the incubator will warm up the plate, and then the moisture that's in the auger will evaporate and will hit the top where the moisture will start to condense. And that would cause a drop to eventually form on the top of the lid. And where is that drop going to drop? It's going to drop onto the plate of media. And if you have cells growing on there, like colonies, the growth will then start merging together. And generally, we don't want that to happen because you just don't want to mix up your colonies, as well as the fact that it doesn't contaminate the culture because the lid of the plate is sterile. However, the bottom part of the edge of the lid is not sterile. I'm pretending this is my Petri dish because I don't have a Petri dish at home. Anyways, if the drop were to come down the edge of the side, the bottom of that edge is not sterile. And then if the drop were to get in the bottom of the plate, it would contaminate the media. And so your risk of getting contamination is higher if you put the plate in right side up and then the drops form on the lid and then drop down. And then it definitely will mix up your culture. And so to prevent that, all the plates that are put in the incubator are put in the incubator upside down. Any question about any of that? All right. So when you get your plate out of the incubator, you may see bumps on the agar surface. Each of these bumps is a colony. Bacteria colonies tend to be smooth and regular. Regular colonies like that's a bacterial colony, very distinct edge, very smooth. And the fungal colonies are not regular on their edge, and they're not distinct, and they're not really smooth either. That's another fungal colony. They tend to be hairy or, oh, what's the other word to use? Hairy or fuzzy looking. The bacteria colonies have distinct appearance. Any question about any of that? Each colony is made up generally of tens to hundreds of millions of cells. To be able to see a colony, the minimum cells you can have is 1 million. And let me see if I got a small one. There's a real small one. There's another small one. So these colonies are at least 1 million cells. And they're very tiny. Most colonies are 10 to 100 million cells. Each colony could be started from one single cell that went through cell division, binary fission specifically, where one cell splits into two, and then two into four, four into eight, eight into 16, etc., until you get 1 million cells, and then you can see the colony. Now, each colony does not have to begin life from one cell. In fact, when I'm looking at this plate here, I would bet you anything that this colony was formed by a clump or at least two cells because it's larger than all of these colonies as well as larger than these. And all of these colonies look like they are the same species that generated the colony. And that is, they look similar. So my guess is these are all formed from the same species. Why I think that one is formed from a clump is it's just much bigger than that one. Now, you can't entirely go by the size to determine how many cells form the colony, and you never will know how many cells form the colony. But generally, it'll be one, two, or a clump that form a colony. It would never be like hundreds of cells because it wouldn't grow in a colony. It would grow like a streak or something. These cells are a little bigger than these cells, and these are the smallest. Whenever you have colonies very close together, They're competing for resources, and so those colonies tend to be smaller than colonies which are somewhat isolated, away from other colonies. So my guess is this is simply formed by one, and all of these are formed by one, and the difference in size is just these are all close together and competing. That one is actually fairly close to this one, but it's the largest of all of these colonies. And so my guess is that one was formed by a clump. But I obviously can't know that with 100% certainty. Any question about any of that? All right, here's a little video for you to watch on binary fission. It's probably very short, so why don't I go ahead and just display it. 16 seconds.
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in two. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
Any question? I should mention that when you're growing your cells in a broth culture, you cannot see colonies, because in a liquid culture, all of the cells just mix together and then grow in the liquid. So there are no colonies in a broth culture. You have to be working in a solid culture before you can get colonies. And usually you will get the colonies on a petri dish, because it's kind of hard to get colonies on a deep or a slant in a test tube. When you're looking at colonies, it's obvious that you can see visible colony morphological differences. And these can be differences, especially between different species of bacteria. When you look at a colony, you can see different colony characteristics, like the shape. The shape could be circular, rhizoid, irregular, filamentous, or spindle. The margin is the edge of the colony. So a circular colony will have an entire edge. A different colony, like an irregular colony, can have the edge either undulate, lobate, or curled. And there's also rhizoid and filamentous colonies where the edge is rhizoid or filamentous. You'll be happy to know I'm never going to ask you to tell me, is this colony rhizoid or filamentous? I'll also never ask you, is this colony undulate, lobate, or curled? Because it's just too difficult trying to separate those colony characteristics. However, I could ask you, is the colony undulate, lobate, curled, or is the colony entire? Because you should easily be able to notice an entire margin compared to one that's undulate, lobate, or curled. And you should also be able to distinguish an entire margin from rhizoid or filamentous. Any question about any of that? All right, colonies also have an elevation. You can have a flat colony, a raised colony, a convex colony, a pulvinate colony, or an umbunate colony. An umbunate colony looks sort of like a mountain. To see the elevation, you should take off the lid of the plate and then look at the colony from its side. And then you can see the elevation. When you're looking at the colony from its top, you cannot tell the elevation. Like here, we're looking at the colonies from the top, and it says colony A is polvinate. I can't see that here. Colony B is flat, and I can't see that there either. And then colony C is convex. I don't really see any difference between A, B, or C. And that's because we're looking at the colony from the top. To see the elevation, you need to look at the colony from its side. You can discuss the colony's size. A punctiform colony is a pinprick size colony. Small is a colony about the size of a pen or a dull pencil. Moderate is something in size between the pen and the eraser edge, or the dull pencil and the eraser. So a moderate is something in between that size. Large is anything larger than the eraser on a pencil. Colonies can be smooth or rough. They can also be glistening or shiny, or they can be dull. Colonies can be pigmented. To be pigmented, the colony must have a color like purple, red, or yellow. Non-pigmented colonies are obviously white, but they also are off-white, tan, and cream colored. Colonies can have an optical property. Most colonies are opaque. You cannot see through them. A few colonies are translucent. And then very rarely, you may see a transparent colony. I've only seen a transparent colony maybe 2 or 3 times in my life. And I'm pretty old. Let's take a look at this plate here. Let me blow it up a little bit. Colony B is opaque. So is this colony here. You can't see through them. This colony there is translucent, and that one there is probably translucent as well. There are no transparent colonies on this plate. Transparent means it must be as clear water. Translucent, you can see through. I'm talking about the edge there. The center is probably another colony growing on top. That is opaque, but right there it's translucent. You can see a little bit through it. Not 100% clarity, but you can see through it. Translucent. All right, any question about colony characteristics? When you're talking about colonies, there's 2 terms you need to know. The abundance. That is simply the total number of colonies on an agar plate. All you have to do is do a count, and you get the abundance. I'm not going to count all of them, but I'll count all of them above this line here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So the colony abundance above this line is 13.
Why not count all of them?
You could, it's just going to take me too long, and it'd be kind of difficult counting the colonies in this region here.
Okay, so just as a reference point for the lab exercises that we're doing after this, there's one that's asking about the colony on the plate. Are we
Abundance,
just
yes.
above the ruler or?
No,
on-
you count all of them.
Okay.
I'm just giving an example, and
Oh,
I
gotcha.
didn't want to try and count all of them
Makes
on
sense.
the plate. It's just too many for me to count. It would take too long. And like I said, I can't really count them in that region, and that region, and this region here too.
More as
You
an
can
example.
count those and those, but sometimes you can't make out the different colonies. Like, I didn't want to count this plate because there's just too many. All right, the diversity is the number of different colonies. So up here, the diversity of these colonies is probably 2. One for this, all of these, and probably that one, although that one I'm not 100% certain. And then that one is definitely a different colony. Yeah, that one's probably different too, but it's at least 2, maybe 3. Okay. And when you're giving the abundance and diversity number, as long as you're close, I'll accept that as a correct answer. For example, if some people said above this line here that there's 2, I would say fine. If other people said no, there's 3, I'll say fine. However, if you say something like 7 or 10, I'd say that's too many. So that would be incorrect. Any question about any of that? Okay, here's a real world example to practice. In this image here, what is the abundance of wild cats? How many wild cats are in this image? Somebody want to guess? All right, work on that. What?
7.
Yes, 7. 2 lions and 5, 1, 2, 3, 4, 5 cheetahs. So the abundance of wild cats is 7. And what's the diversity of cat species?
2.
2, the lions and the cheetahs. Good job. All right, know the terms of this lab. There's a cute image of somebody's hand. And now let's go to the worksheet. You're not actually going to be doing the lab, but we're going to supply pictures for you to run through the lab and do the lab. In lab module 1, ubiquity of microorganisms, we're going to look at procedure 1, where we're going to ask the question, what is in the air you breathe? And what we did was we exposed the plate to the air for a given amount of time. And my first question for procedure 1A is, what is the colony abundance on this plate? Just count the number of colonies. And as long as you're close, I'll accept it. But if you're far off, like more than 5 away, then I will not accept it. And you'll get marked down for that. But remember, in the lab, you can turn in twice. Procedure 1B, what is the colony diversity on this plate? I don't want to answer for this, but we've already answered that in another plate. And then procedure 1C, what steps could you take to reduce airborne contamination while you are inoculating a plate? So don't tell me a step you could take before you inoculate the plate, or tell me a step you could take to reduce contamination after you inoculate the plate. Tell me while you are actively inoculating the plate. Procedure 2 is looking at what is on the table, the lab bench. And what we did was we sampled the lab bench before we disinfected it. And there we got the growth from the table before disinfection. And then we disinfected the table and then sampled the plate And this is the growth we got. There might be one colony right there. There could also be an air bubble. It's kind of hard to tell here if it's a colony or an air bubble. If you were to really look at that in real life, you would know right off the top of your head. And then answer procedure 2's 2 question. Procedure 2A, did table disinfection work? It's simply yes, no, although you can attach an explanation if you wish. And then procedure 2B, what's the purpose of allowing the table disinfectant to air dry? Or when we disinfect a table, why do we air dry the disinfectant? So answer that question and make sure you're answering it correctly, whether it's during the inoculation or some other time. Procedure 3, what's on your hands? What we did in procedure 3 was we touched the agar plate with our fingers before we washed our hands. And then we washed our hands, dried them, and then touched the agar plate again. And as you can notice, after washing your hands, there are fewer organisms, but there are some organisms after washing your hands. And the first question is, does hand washing work? Also, yes or no, although you can explain it. Optionally, you can explain it. And then procedure 3, number 2, suggest one reason why hand washing did not result in the complete removal of microbes. Hint, it may have to do with the normal microbiota on the skin. Note, if you obtain an answer following research, please provide a citation of your information and your information source. Procedure 4, when you kiss your significant other, what exactly are you giving him or her? Somebody has touched the lips with the plate, giving these patterns. And on this plate, somebody has touched their tongue on the plate, giving this pattern. Now you should note, I've added a note about these two plates and what you're seeing. So you should read that note. And then which plate has the higher colony abundance? The tongue plate or the lip plate? And answer that. And then procedure 4C, why was the tongue plate incubated at a higher temperature than the lip plate? If you don't know that off the top of your head, you'll have to do some research because that one would be a little tricky to get. This one, most people do get it wrong the first time, but it isn't that difficult once you at least understand what's going on. And then procedure 5 are microorganisms in the soil. What we did was we took a little bit of soil and then put it in a tube of water. We then inoculated that tube of water onto a plate, getting this soil bacteria growth on the agar plate. And then answer the questions. Procedure 5A, what's the colony abundance? Is it even possible to accurately answer this question? Please explain your answer. And then procedure 5B, is there greater abundance of colonies from the soil compared to the lip plate? And occasionally I get students writing, well, the lip plate has small colonies, and the soil plate has huge colonies. And I'm going to argue that that is not correct. First, let's go talk to the lip plate. You can see some very large colonies that were obtained from the lips. There's one there, one there, one there, and one there. And those large colonies all have color too, except for that one.
On the lips, are the surrounding, like on the top and bottom of the lip, are those really small colonies?
You're talking about like this one here?
Yeah, like the outer rim where you see all the
Yeah,
little dots. Are
those
those
are
small
colonies.
Okay.
They're just smaller ones. This one probably has about 1 million cells, and that one much more than 1 million.
Okay. Thank you.
All right. So procedure 2B, is there a greater abundance of colonies from the soil plate compared to the lip plate? I think I had started this discussion, and I did mention that the lip plate doesn't entirely have small colonies. We definitely have a few very large colonies. This one, 2, 3, 4, 5, 6 large colonies. And it is true the soil has more large colonies than 6, but it's not as many as you might think. Meaning all of this region here, and this region here, and probably that region there, and this region here, are formed from small colonies, not from large colonies. So there are a number of small colonies on this plate, and I'm going to guess it's close to 50%. If it's not 50%, it might be a little bit under 50%. All right, any questions about the lab?
I have a question about what we were just looking at. And I don't want to like give the answer away, so I don't know if I should ask you later, but I'm just having a little bit of a hard time distinguishing on this one what's colonies and what's not, because you can kind of see the distinguished margins, but then it just kind of there's kind of big clumps there that don't look like colonies, but then I don't know if that's a bunch of
Oh,
colonies
you're talking
together.
about these clumps here?
Yes. Yeah,
Those
exactly.
are colonies. And there's so
Oh,
many of them there that instead of having individual colonies, you're getting the growth in a clump. And this clump
okay.
in particular, that looks like a different colony. That one looks like it's got orange in it. That one looks like a different colony. So this one has at least 4 or 5 different species making up this clump.
And does the whole surface area of that clump, is that all made up by colonies? I guess that's
Yes, this whole area is made up by colonies, not one,
okay.
but
And
many. I have no idea how many.
okay. Thank you so much.
Uh-huh. All right. Any other questions? If not, I'm going to stop the share and stop the recording. All right, there we go. So a student has asked a question about lab exercise 1. So you're talking about procedure 1. And which question?
B for the colony diversity.
What is the colony diversity on this plate? All right, so we're looking at this picture here. All right, which colony are you having a hard time
Well, a
deciding?
couple, I'm just making sure I got it right here because a lot of them are different shades of similar colors. So should I count different diversities by the different colors? For example, if you look above the 2, just above it, there's like a yellowish, and then there's more of a grayish whitish. Would those be considered 2 different?
These two would be considered 2 different.
Okay.
Okay. This one and that one are probably all the same. Even though this one's much larger, it was probably formed by a clump.
Okay.
question is, I'm not sure if those two are the same or not. Let me blow that up. Maybe they are the same.
Is
Anyway,
there some leeway with this
like
question?
I
just
said,
like
you'll
with
get
weight?
some leeway. How many colonies did you get for your abundance?
Oh, for me, I wrote 28.
So close to 30.
Mhm.
So yeah, as long as your number is within 5 or 6 from what I estimate, I'll accept your number.
Okay, sounds good.
Okay.
Can
Thank
I
you.
ask a really quick question too about the same photo? Is it possible to have one colony growing on top of another colony?
I bet you're talking about this one right here.
Yeah,
That one, yeah, that's
exactly.
2 colonies. That's the yellow one and that's the brownish one.
Okay, great to know. Thank you.
Oh, let's stop sharing and stop recording.